Welcome back to this YouTube channel. For new viewers, kindly subscribe and also don't forget to click the notification bell so you get notified each time a video drops on this page. Also, don't forget to check out our previous video on microscopy. Today we'll be looking at the photographic recording of ultrastructural images. Let's get right into it. Introduction. An electron microscope is a microscope that uses a beam of accelerated electrons as a source of illumination. It is a special type of microscope having a high resolution of images. The electron microscope uses signals arising from the interaction of an electron beam with the sample to obtain information about structure, morphology, and composition. The electron gun in an electron microscope generates the electrons. To move electrons down the column, we have an accelerating voltage of 100 kV to 1000 kV is applied. The denser regions in the specimen scatter more electrons and therefore appear darker in the image since fewer electrons strike that area of the screen. Then both formaldehyde and butyraldehyde, a fixative often used for electron microscopy, react with the amine groups of tissue proteins, preventing their degradation. The electron beam coming out of the specimen passes to the objective lens, which has high power and forms the intermediate magnified image. The ocular lenses then produce the final further magnified image. What are the types of electron microscope? We have the transmission electron microscope. The transmission electron microscope is used to view thin specimens through which electrons can pass, generating a projection image. Then it is analogous in many ways to the conventional light microscope. The transmission electron microscope is used, among other things, to image the interior of cells in thin sections, then the structure of protein molecules, also the organization of molecules in viruses and cytoskeletal filaments. Other infiltrating media are the paraplast, the gelatin, collidines, the epoxy resin for electron microscopy, and the acrylic resin for immunohistochemistry are used. The scanning electron microscope is another type of electron microscope. It's a conventional scanning electron microscopy. Conventional scanning electron microscopy, it depends on the emission of secondary electrons from the area of a specimen. Then because of its great depth of focus, a scanning electron microscope is the electron microscope analog of a stereo light microscope. It provides detailed images of the surfaces of cells and whole organisms that are not possible by transmission electron microscope. It is termed the scanning electron microscope because the image is formed by scanning a focused electron beam into the surface of the specimen in a faster pattern. That the application, the electron microscopes are used to investigate the ultrastructure of wide range of biological and inorganic specimens, including microorganisms, cells, knowledge molecules, viruses, samples, metals, and crystals. Industrially, the electron microscopes are often used for quality control and failure analysis. The modern electron microscope produces an electron micrograph used to utilize digital cameras and frame grabbers to capture the image. What are the advantages? They have very high magnification, incredibly high resolution as well, and also materially barely distorted by preparation. Then it is possible to investigate a greater depth of the field. Thank you guys for listening. That's all for now. Please don't forget to keep in touch so you get notified each time we drop a video. Thank you for listening.